can see the enzyme in the blood. Normally, the enzymes are not synthesized or not directly produced inside the blood, but we get enzyme in the blood because because the cells may rupture the cell membrane and have a particular because of that they come into the blood. Okay, these are the conditions which affect the leakage of enzymes from hypoxia is a decreased oxygen inside the hello. Can you hear me? Tell me if you can hear me. Okay, so hypoxia means decreased oxygen uh, level of oxygen inside the tissues. Okay, so hypo means less and oxia means oxygen, so less oxygen. So less oxygen in tissues and cells usually occurs because of the result of thrombosis. Thrombosis means the clot formation. There may be another reason like cardiorespiratory failure. Maybe the blood is not able to be pumped from the heart, so cardio failure or respiratory failure or cardiorespiratory failure. And the third cause can be the anemia, where the hemoglobin is less, RBC is less, and because of that, the cells may not be getting appropriate amount of oxygen inside the cell. Next is the chemical and the drugs. Now, these chemicals which we take from the outside, the drug that we take from the outside, especially lead, mercury, and alcohol. So they causes leakage of enzymes from the cell. Physical agents such as trauma, radiation, and electrical energy. So these agents may can cause trauma into the the tissues or the cells, and uh, because of that, there may be the cell damage or cell membrane damage, and it result into the release of the enzyme from the cell to the blood. Microbiological agents, the very common like viruses, bacteria, fungi. If you have infection with this microbiological agent, then usually your cell may get ruptured, your cell may get inflamed. So inflammation of the cell and tissues can result into the leakage of enzyme from the cells. Immune mechanism, this is a hypersensitive reaction or anaphylactic reaction or cytotoxicity, immune complex disease, cell-mediated hypersensitivity. Here there is a binding of antigen with antibodies, and this antigen-antibody reaction may result into the cell damage, cellular damage, results into the leakage of enzymes from the cell to the blood. Some genetic defects like diabetes mellitus or maybe the GOT, which can increase the release of enzymes from the cell. Next is a nutritional disorder, there is a malnutrition like condition, protein, calorie malnutrition, or vitamin deficiency or mineral deficiency. Because of this nutritional disorder, there may be the lack of the nutrients in the cell, and because of that the leakage of the enzymes may be more than the normal condition, and usually there will be the more enzyme in the blood because of this nutritional disorder. So there is a leakage of enzyme from the cell in whatever the uh, we have studied the various reasons, various causes that can increase the leakage of enzyme from the cells. Next, we can see here, this is the image which is showing that this is the normal cell turnover, and this is the cell which is which is showing necrosis. So cell necrosis and the cell, this is normal cell turnover. What is normal cell turnover? Normal cell turnover means the normally cell has a limited lifespan, and because of that, after completing the lifespan, usually cell dies, and the cell enzymes usually mix into the blood. So the enzymes, when it mix into the blood, we can be able to detect the small amount of enzyme into the blood by doing the enzyme test. So this is normal cell turnover, but this cell turnover may be increased when there is a cell necrosis process, the pathological process, where the cells has enzymes, and the cell necrosis means cell damage, cell destruction can increase the excess amount of enzyme secretion into the blood. And when we take the sample from the blood, and you will be able to find that there is increased plasma level of intracellular enzymes. So enzymes level may be increased into the blood, and this the increase is because of cell damage, right? So in tissue damage or necrosis resulting from injury or disease is generally accompanied by increase in the level of several non-functional plasma enzymes. So actually, these are the non-functional plasma enzymes. Why we say non-functional plasma enzymes? Because these enzymes are normally not present in the plasma. They are usually found in the cells. And when there is a cell damage, so cells damage may release the enzymes into the blood, and after that there will be the excess level of the plasma enzymes here, and this is what can act as a marker, disease marker. Okay, so enzymes as a diagnostic marker can help you to diagnose a specific tissue damage or a specific organ disease. Altered enzyme production is another way, which this let's know what are the altered enzyme production. The enzymes are produced inside the cell, okay, living cell, and these living cells have a message, the DNA, the genes, and because of the genes, the specific enzymes are produced inside the cell. And process of the enzyme production is what the transcription and translation process, and the enzymes are what they are produced in the cell, and in this production involve the translation process with the help of ribosomes and Golgi apparatus in the plasmic reticulum, so we get the functional enzymes here. So there can be the decreased formation of the enzyme production, the genetic defect, or it can be acquired. So there can be the deficiency of the gene, or there can be the disease in the genetic disease. Because of that, there can be the decreased formation of enzyme, or there can be the acquired reason, means after birth the gene is normal, but even though it can increase, it can alter the the enzyme production. Okay, so there may be some defect with the ribosomes, there may be the defect with the endoplasmic reticulum, there may be the products defect with the translation transcription. So okay, so decreased production of the enzyme, so this is what it can be acquired or it can be genetic. Next, it's increased level of enzyme because of increased production. So normal growing children or end of normal pregnancy or disease. So these are the conditions where there can be the increase in the enzyme production. The growing children age has an increased enzyme production. At the end of the normal pregnancy, there can be the increased enzyme production, and at the disease level also, there is increased enzyme production. Enzyme induction, the enzyme which are produced, so it can be there will be a stimulation process which can induce the enzyme to produce more and more. So there can be the drugs that you take, it can be the alcohols, or it can be the biliary obstruction which make an induce the production of enzyme from the cell, and because of that more enzyme production, so there will be more enzyme in the blood. Cellular proliferation is another way, which is a neoplasia, which is like a cancer. So increase in tissue source of enzyme as in malignancy, where the tissue source or cells number, cell population is higher than the normal, and because of the neoplasia, increased tissues mass, there can be the more increase in the enzyme level in the blood. Similarly, increased production of enzyme within the cell resulting in increase in serum by overflow. So this increased production of enzyme within the cell is maybe because of some stimulant, or maybe because of uh, some pathological factor which can induce the enzyme to flow, increase the enzyme to produce more in number. Okay, and because of that, there will be the increase in the level of enzyme in the serum, and this is what we have seen, that the increase production of enzyme in the cell and result in the increase in serum by overflow. Next, necrosis of cell. So cell necrosis. What is the cell necrosis? It's the cell damage, cell destruction. So if there is cell destruction, okay, maybe because of the microbial agent or maybe some immune mechanism, so or so that can leads to increase leakage of enzyme, in the cell. And last, increase permeability of cell without gross cellular damage. So permeability of the cell membrane, if it is increased, so it can leads to increased level of enzyme in the serum. Enzyme inactivation. So this is another factor which can alter the level of enzyme in the serum. So enzyme inhibition is one of the factor. Like let's say it's a reversible inhibitors and irreversible inhibitors which can bind with the enzyme and can inactivate the enzyme activity. So this inactivation of the enzyme can, if you stimulate the enzyme in the serum, and because of enzyme inhibition, so you will not be able to find the appropriate amount of enzyme in the serum. Lack of cofactors like such as NAD, FAD, magnesium ions. So they are cofactors. If there is a deficiency of these cofactors, and then it leads to you know uh, enzyme inactivations. So enzyme, if it is inactivated, so you'll find that there will be a less number of enzymes, even though the enzymes are produced in the large quantity, but you'll see that the enzymes are at the less in number. So this is what we have studied about the enzyme, uh, about the enzyme, the factor affecting enzyme level in the serum. So these are the different factors we have studied that affect the enzyme activity, enzyme level in the serum. Now we can discuss about some questions if you have, and we can try to clear the doubts here. We have come to the end of the questions now. Any questions?